Welcome everyone, and thank you for joining us for today's webinar titled Studying Retinal Function in Large Animals, Laser-Induced Choroidal Neovascularization in Pigs. This is Martin Hess from Inside Scientific, and I will be your host for today's event. We are happy to share that over 100 scientists from around the world have registered to participate in this webinar, and I'd like to personally extend a warm welcome and thank you to all of you who have taken time out of your busy day to be with us. Our session today is sponsored by Phoenix Research Labs, whose mission is to develop technology for eye and eye-brain research using animals. Our presentations will focus on the Micron X large animal camera that captures high-resolution, detailed images of a range of large animal species, allowing bright field and fluorescein angiography insights into your research. First, we will hear from Christine Van Hover from Phoenix Research Labs, who will introduce us to the Micron X large animal retinal imaging system. Dr. Van Hover will discuss how to take high-quality images of the whole large animal retina, what species can be imaged with the Micron X camera, and show examples of fluorescein angiography in non-human primates. Our second speaker today is Dr. David Culp, Director of Research at Powered Research. Dr. Culp will present data from a recent study designed to evaluate and characterize a porcine model of laser-induced choroidal neovascularization, or CNV. Dr. Culp will provide specific examples of his data where fundus photography, fluorescein angiography, and retinal optical coherence tomography were performed. He will also show how the Micron X large animal imaging system produced high-resolution fluorescent images to quantify neovascularization.
Thank you for that introduction, Martin, and thank you everyone for joining us for the webinar today. As Martin said, my name is Christine and I'm the Chief Scientist here at Phoenix Research Labs. That means that I get to test out the equipment we make here in our lab, and I also travel around the world to train scientists on how to apply the equipment to their research. So today I'll be introducing the Micron X large animal camera to you, and I will go over what Phoenix Research Labs does and share some beautiful images from our newest offering, the Micron X. So Phoenix Research Labs makes, designs, and manufactures retinal imaging cameras. Our primary offering right now that you may be familiar with is the Micron 4, and that is a fundus camera for rodents, primarily mice and rats. So on the right-hand side of the screen at the top, you'll see that a fluorescein angiography image of a rat. And so this is a typical image of fluorescein angiography. You can also get bright field images as well as other different fluorophores. Attaching onto the Micron 4, we have some offerings like the OCT, which is our optical coherence tomography, and that allows you to look at the structure of the retina without cutting into the animal. These are all done with live animals. So on the right-hand side, that middle image is a typical OCT image, and at the far right, there is what looks like a tumor, so retinal blastoma forming. So you can see the tumor developing throughout the development of this animal and examine it in a longitudinal manner. We also have electroretinography, or ERG, that measures the function of the retina. So on the right-hand side at the bottom image, that's the readout that you would expect from an ERG recording, flashing light into the eye and recording the electrical responses. We also have anterior segment slit lamp, a Ganzfeld full field ERG, and a laser that induces CNV, or choroidal neovascularization. So these are all for rodents, as I said, and these are in labs around the world, but we haven't had anything to study large animals up until now. But we introduced a separate system, separate from the Micron 4. This is the Micron X, and it's optimized for large animals. It has a wide angle 100-degree field of view, a resolution of about 8 microns, and has bright field and fluorescein angiography capabilities. So on the right-hand side, you can see that these are some pig images that David will talk about in more detail later. On the left-hand side, we have bright field images, and on the right-hand side, we have fluorescein angiography. So why study large animals? They are a critical step between rodents and clinical human trials. Rodents are great for a relatively low-cost and high-volume method of testing different drugs and disease development, but they are obviously quite different from humans. So larger animals have a similar eye size to humans, and you can test how the drug efficacy or disease model works in a different species. And you can also do things like allergy testing in species such as guinea pigs. So guinea pigs will have an allergic reaction similar to many things that humans are allergic to. And so you can test, for instance, your drug in a guinea pig eye and see if it has an allergic reaction, then you probably don't want to test it in humans. On the right-hand side of the screen, this is a picture of the actual Micron X. So it's a small, compact, and portable system. On the bottom, there's a control box, that white box, which everything rests on. On top of that is a laptop, and this will have our custom software that will have a live video feed of whatever is being imaged. The handpiece, or the imaging handpiece, is resting on top of the laptop, and this is what you will actually, it's a handheld imaging system, so you'll hold that blue part and touch that to the eye, which I'll discuss in the next slide. And there's also a foot pedal, which isn't shown here. And you can adjust the brightness and the focus with both the foot pedal and the control box. So using the Micron X is a simple 4-step process. First, you sedate the animal with either isoflurane gas or with a ketamine solution, and you then dilate their eyes. We've also found that using a speculum in that to keep the eyelids out of the way, especially with isoflurane, is useful. You then position the animal and apply gel onto their eyes. The position of the animal depends on what species you're using. So I've found that using non-human primates, they lie on their back and look up at the ceiling, and then you image from that position. Rabbits, who have eyes obviously more on the side of their head, it's most useful if they're lying on their side and again looking up towards the ceiling. And David and I experimented with pigs, and we found that that works best if they are actually lying on their stomach and you approach them from the front rather than them looking up at the ceiling. So once you apply gel, the gel is going to be goniosol or something similar, and that is an optical imaging medium as well as protection for the eye. You take that Micron X handpiece, which I showed you in the previous slide, and touch the lens onto the eye. And this way you get these really clear, crisp images. You adjust light level, focus, and gain. So light level and focus are adjusted manually through the control box or with the foot pedal, which is a really easy foot rocker motion. You can have, I've found that you can either have one person imaging using all these controls, or you can have two people where one person holds the handpiece steady onto the eye and the other person adjusts the controls. The gain is controlled through the software, and you can take images with the foot pedal or with the software. So once you have all of these factors adjusted and you have a nice image that you are pleased with on the live video screen, you can take still images or video, and you can move the Micron X around the retina and get different angles and take images and videos all throughout these points. So now I'll show you a bunch of different images, examples that I've taken in a range of species. So on this slide we have a cat retina, a pig retina, and a dog retina. So the cat and dog both have these hyperreflective cups at the top of their retina, and so I've adjusted the light level to have that in clear focus. You can also increase the light level and have that darker bottom part of the retina more illuminated, but the hyperreflective cup will become a little bit bleached out. So you can take two images with a lower light level as well as a higher light level. So you can see the stark contrast of the vasculature against that hyperreflective cup, as well as the details of the green and blue colors that are in the retina. So it's a really nice image there. And the pig retina in the middle is a different sort of pig than the ones that David uses. You can see that you're getting really nice illumination and focus of the retina and the vasculature. So those are all images of animals who have darkly pigmented retinas. The really nice thing about the Micron X is that we have imaging versatility, and you can get crisp images of both light albino retinas and darkly pigmented retinas. Sometimes fundus cameras can struggle with the more darkly pigmented retinas, but Micron X gets really nice images of those. So on this slide, on the left-hand side, we have an albino rabbit retina. This is pretty typical, this stripy looking retina here with the vasculature going out horizontally. And so obviously albino means there's not a lot of pigment in the eye, and you can see that you have clear illumination of the vasculature and the retina with an albino animal. So this is a lightly colored retina, really good illumination. In contrast, on the right-hand side, we have a very darkly pigmented macaque non-primate retina. And so I have two images here because I have slightly different light levels for these two. So on the top, we have a slightly lower light level, and you can see if you look at the optic nerve, which is on the left-hand side of the top primate image, you can see the vasculature going into the optic nerve area. You can also start to see the nerve fiber layer. So that on the top of the optic nerve, there's that sort of shiny streak, and then also to the right of the fovea, there's something that to me looks a little bit like an oil spill, an oil sheen. That's the nerve fiber layer. The retina is still well illuminated, and you can see the vasculature, and you can also see the nerve fiber layer sort of on the blood vessels on the bottom as well. In the bottom image on the right-hand side, that is a slightly higher light level. And so you can see that the optic nerve is a little bit more bleached out. You don't get as much detail, but the fovea and the darker areas of the retina are have a little more detail in them. So this is really easy to do while you're imaging the animal. You just increase the light level and snap some images, and you get totally different information, and that's really nice. We also have stunning fluorescein angiography ability. So this is again a macaque non-primate retina, and on the left-hand side we have that bright field image, which you've seen similar images to this, and on the left-hand side, the right-hand side, we have the fluorescein angiography of this same animal. So with the Micron X, you can pop out the white LED for the bright field and then put in the blue LED, which will cause the fluorescein to fluoresce green, and there's a simple switch to switch the imaging filter over. So while what we had is that the primate was sedated, looking up, we took some bright field images, and then as the IV was being prepared and the fluorescein injected, I switched over the LED really quickly and then got this fluorescein angiography image. So I'm going to show you some my favorite images I've taken since I've been at Phoenix so far, and these are some videos of the fluorescein angiography. So what we did is I had the Micron X up to the eye, we had the blue LED in, the filter switched over, we pushed the fluorescein angiography, the fluorescein into the veins and took a video. And so what we'll see here is this is a real-time video, so it's the full speed, and the next slide will be a slower video. You can see the arteries filling up first. Again, you can see the arteries filling up first with the fluorescein, and then they dim while the veins fill up with the draining the fluorescein back to the heart. So, and then the capillaries also fill up. So I'll show this one more time. You can see the heartbeat rate as well. Okay, so in the next slide, it's going to be a slowed down version of that. So what I want you to notice is, again, the arteries filling first and the vein, then the veins draining, and also you can see the heartbeat in the first few seconds. So switch over to, here's the slowed down video, so you can see the heartbeat suddenly fill in the veins, which is really cool, or excuse me, the arteries. And then right about now, the veins start to fill up. And all through this, the capillaries slowly become more and more illuminated. And this is a video while it is a handheld system, and so you can see that this is about the amount of shake that you can expect, which is, and it's not a lot of movement happening. So there's a bit of eye movement, and perhaps my hand was moving a little bit, but for a handheld system, it's still really steady, which is nice. So again, this is my favorite video I've ever taken in my tenure here. It's just beautiful seeing everything, all the vasculature fill up with the fluorescein. Okay, so this is a time lapse. So it was the same video as before, and what you can do is take the video and have the timestamp of when you injected the fluorescein and have that timer running, and then you can take still images from the video really easily in our software. So going from left to right, you have the early time point of about 16 seconds after injection, and you can see that there's the major arteries are filled up, but not a lot of the capillaries. And going over to the right side, you can see that the vasculature gets more and more filled up and illuminated. And so this was really easily done. You just take the still images, you play the video, you pause it, take the still image out, and here you have both the video and now you have these still images, both of which are easy to present. So final discussion or final images that I have, and David will be talking in much more detail about this, we have the CNV induction in pigs that David did at Powered Research. So again, examples of a bright field on the left-hand side and the fluorescein angiography on the right-hand side. And these are with pigs, and David did some laser shots in the pigs and looked at some time points. And you can see some CNV induction happening on the right-hand side. With the top group of pigs, he injected them with saline, so essentially no treatment, and you can see that there's clear CNV development or choroidal neovascularization on the right-hand images. But in the bottom set of images, he treated the pigs with Eylea, which is a treatment against CNV that's used in humans as well. And you can see that the laser shots in the bright field look much less bad on the bright field, and in the fluorescein angiography you can see that there's really a significant decrease in CNV development.
Well, thank you again everyone for participating in this webinar, and Christine, thank you for that introduction, and Martin as well. My name is David Culp. I'm the Director of Research at Powered Research, and what I wanted to talk to you about today was a model that we've been developing and running here at our facility. It's a laser-induced model of choroidal neovascularization in swine. And we wanted to evaluate a current standard of care, aflibercept, in this model to evaluate how robust this type of model could be to test other therapeutics targeting macular diseases. So a little bit about Powered Research first. We're a preclinical contract research organization based in Research Triangle Park, North Carolina. We provide high quality and cost-effective non-GLP studies, and about 80% of our business is in the ophthalmic space. We do some orthopedic and oncology work here, but again, we specialize in ophthalmology. Not only do we run a number of efficacy models here, we also do safety and tolerability assessments of drugs and devices and other drug formulations. But we've also found that our clients also have a need for efficacy models to test their therapeutics, and so one of the things that we thought about is of course there's a lot of focus in macular diseases, and really what you want is a larger eye to be able to test your compound in. So moving on, there are several preclinical models of choroidal neovascularization that are out there, and I'm sure many of you are aware of them. But they have limitations, and pharmacokinetically, rodent models are not robust because it simply is just such a small eye, and you can't dose a volume that you would dose in humans in rodents. They're fantastic for getting an early read on a compound or screening analogs of compounds or narrowing down new chemical entities, but they really don't provide a system for being able to evaluate what happens in a larger eye. Other models that sort of look at leakage or vascular disease in the eye are the VEGF-induced models. For example, in the rabbit, you inject recombinant human VEGF 165, and within about 3 days that protein elicits leakage in the medullary arrays. And while it's a very robust model that again allows you to screen compounds and look at different analogs, it's really not in the right part of the eye to study choroidal neovascularization. And then finally, non-human primates are probably the gold star here, but they're certainly very expensive, and these are usually for compounds that are further down the pipeline and are going into GLP and pre-IND filing. So A lot of times companies come to us don't have that kind of money to be able to run these types of models. So with those limitations, we set out to sort of set up a model of choroidal neovascularization in the pig. And so one of the advantages of that is that the pig eye has a very similar architecture to it, to the human eye, probably without having to go to a non-human size eye to work in. You can inject 50 to, we've injected upwards of 150 microliters of volume in the eye. While that might affect intraocular pressure, we don't see much efflux on those injections, so the eye can handle that. We use weanling swine, so we use just regular farm pigs. There's plenty of those in North Carolina, which is fantastic. And with that, that actually helps us reduce costs. We don't have to go to expensive transgenic mini pigs or other species, so or strains, pardon me. So these are the animals that we use. We use males and females, and we've run the study 3-4 times here at Powered Research, and a number of times in collaboration with Dr. Gilger at NC State University. We use 6 to 12-week-old animals that weigh about 12 to 20 kilograms, and we induce about 5 to 6 lesions per swine eye, equidistant from the optic nerve, and we use an 810 nanometer diode laser to induce those lesions, and that is directed through an indirect ophthalmoscope. So this is an image, these are some images of the lesions that we induce in these animals. And so on the top panel, what you can see are balanced salt saline injected eyes, and on the leftmost, this is just the ocular fundus of these animals. What you can see immediately post lasers that we have 6 nice uniform laser spots that have been induced in these animals. Don't really see a big difference in the fundoscopic images across the board of these animals. Potentially some differences can be highlighted at 3 and 4 weeks, but again, this is just to demonstrate and show that these are very consistent lesions. They're all about the same size. They don't interact with one another, and I can talk a little bit about that later. But again, this is what these lesions look like. So what I wanted to do next was kind of show you how we do an intravitreal injection. So these animals are brought into our facility and acclimated for a minimum of a week. Our technicians spend time with them. They're group housed. It helps to get them familiar with you so that you can handle them a little bit better. And so what we do quite often is in some of our studies is we will laser the animal and then inject immediately after we laser the animal while it's still tranquilized. Some protocols warrant that we laser and then a week later we dose. Again, a lot of those things are dependent on the sponsor's compound and the mechanism of action and really what biology you're trying to affect. But again, in this particular study, what we did was we lasered the animals. The animals then were taken into the prep room and given a sterile prep with betadine and water rinse to really clean the eye up. Again, these are pigs, so they tend to be messy, so you really got to make sure you clean the orbit very well before you do an intraocular injection. But what I wanted to show you was how we do an intravitreal injection. We use insulin syringes and we inject typically 50 microliters. It's well tolerated, but I said you can go up to 100 without really much problem. So the procedures actually are very fast. We're able to the tranquilizing the animals actually takes longer than the actual procedures do. But in this next slide, what I'd like to show you is what these lesions look like by OCT or optical coherence tomography. And we perform these over time as simply to look at to confirm that we ruptured Brooks membrane number 1 and immediately post laser, but then also to look at whether we had any retinal swelling or severe edema or other types of pathology as a result of the energy that we delivered to that tissue. So if you look at the very left, prior to laser, this is just the en-face view on the top, and then of course the cross-section below, and you can see where this green line goes through the en-face view. That's the cross-section of, or the particular section of the retina that you're looking at. Immediately post-laser, which you can see is, as I'm circling here, this is where the lesion was placed. And if you look down here, you can clearly see that we've ruptured Brooks membrane. There's a fair amount of edema around that lesion, as you might expect from the injury. 2 weeks later, there's still some edema present there, but not a whole lot of tissue remodeling taking place adjacent to the lesion. And I'll show in the next slide some quantitative values. We drew calipers just to take a look at that. And then at 4 weeks, again, really not a whole lot of difference in the central part of the lesion or adjacent to these lesions. So again, this is just a graphical representation of that, where we, in both aflibercept and BSS treated eyes, we don't see certainly any adverse effect to the adjacent retinal area adjacent to these lesions, which suggests that they're sort of independent of one another, that we don't have too many in the eye causing sort of collateral damage or other types of healing responses between the two. So we feel like we've got them spaced pretty well. The central lesion retinal thickness does sort of decrease, as you might expect, because we've induced a lesion in that area. But we don't see very large differences again over time between the two treatment groups. So they really sort of were about the same. There's maybe a slight dip in the central retinal thickness in the aflibercept eyes, but certainly not statistically significant. So as we were working on this model, we really felt like we needed a good camera, a good way to sort of image and look at these lesions in these eyes, and it was very convenient that Phoenix came out with this camera, the Micron X, because it's been fantastic in helping us characterize this model and bring this model online. At day 7, what I'm showing you here are some images across the top panel there, the bright field images of some representative BSS treated eyes 7 days post laser. And just below that are the corresponding angiograms. And then of course below that are the Eylea treated eyes at day 7. And I'm showing you this because I hope what you can appreciate in these images, in the BSS treated eyes at day 7, you can really see you've got a lot of diffuse fluorescein signal coming out from the boundary of these lesions. You can also see in this far right animal, you've got some hyperfluorescent areas within the central portion of these lesions. These are suggestive of nascent vasculature and leaky vessels in the choroidal space, really just dumping a lot of that fluorescein out. And what you do not see is that in these day 7 Eylea treated eyes. So what you can see, there's not as much diffuse leakiness around the outside of the lesion, and the central portions of these lesions are dark. There's really nothing, no biological activity with respect to fluorescein leakage in the central part of these lesions. So I think there's, just visually you can see there's quite a significant difference in these two treatment groups. Just a side note, we do all of our angiography on these animals. Once they come into the surgical suite, we place a catheter and we administer the fluorescein and then start the stopwatch, of course. Generally within 1 to 2 minutes, we start our first measurement. We're able to laser both eyes in these studies, so we tend to do our left eye first and then our right eye. Of course, that can potentially skew the data in the sense you might have more leakage in that right eye because there's been more time since the fluorescein's been administered. We've looked at that using the video function with the Micron, and we've not seen significant differences in the short amount of time that we are able to do this. We're able to really capture these images, left eye and right eye, within, say, 1 to 2 minutes max. So again, that's sort of the process that we use. Day 14, the images and the lesions, again, are less hyperfluorescent overall, and I think that's just a normal wound healing response that's taking place. So what you can see, though, and appreciate, I hope, again, you have this very diffuse area around these lesions in the BSS-treated eyes, and looking down at some of the ILEA-treated animals below, it's almost very difficult to identify the individual lesions. So this drug's doing a very good job at blocking that neovascularization. So I wanted to kind of talk a little bit about how we analyze this data. So we take our best image. Usually we collect stills. We don't take videos, simply because it's faster to process the animals that way. But we'll take up to 15 to 25 images per eye, and then one of our scientists will take time going through each one of those to identify the best one that's in focus, that's got all the lesions in it, and just looks the most uniform. And so what we analyze and the way we calculate our signal is using a corrected total lesion fluorescence measurement. And so what we first do is we define an area of interest, or we define the boundaries of that lesion. As, for example, in 2 here, you can see this yellow line that I'm tracing around. So that's sort of the area of the lesion. And then we take an area immediately adjacent to that, because we feel that that area would have a similar background. For example, if I just took a single background image and say I took it down here around 10, the background there is very different than the background up here. So for each of these spots, we normalize it to an adjacent region that tries to capture some of the vasculature, because again, all of that is incorporated into the lesion fluorescence that we get. So, again, to back up, our calculation is an area of the lesion, and then we subtract the mean fluorescence within this background region, and then that's multiplied by that background in that lesion, and that number is then subtracted from the fluorescence within our actual lesion size, and that's how we calculate the fluorescein angiography in these animals. It's a corrected total lesion fluorescence. So what you can see at day 7, and I hope you could appreciate that in the angiography images, you can see that ILEA really inhibited the leakage of these lesions by almost 60%. Typically, I see between 40 and 55%. So this particular study, we saw a really robust effect at day 7. The lesions, as I said, this is a wound healing model, so these lesions do heal. They don't completely resolve. At least we've looked out to day 28, and we don't see that these lesions are gone by any means. But again, the amount of leakiness is most robust at day 7, and we've also looked at day 10, and really that's the peak. But even so, at day 14, there's still about a 50% inhibition of signal, and again, that carries on at day 28. These error bars are large because this was a small N. We only had 2 animals in this particular study. We've also looked at flat mounts in these animals, and so what we do at the time points that are listed here, we enucleate the eyeball and fix it. This is very similar to what you would do for rodent choroidal flat mounts. So you fix the eye, you take an eye cup, remove the neurosensory retina, exposing the retinal pigment epithelia with the choroid and sclera. And then we stain that tissue with DAPI, which is a nuclear stain, phalloidin, which stains F-actin, and isolectin IB4, which is the blood vessel and microglia marker. And what we quantify is that IB4 signal, and we use a fluorescent microscope and software to be able to evaluate that. And what you can see here is at week 2, ILEA is having some effect on these lesions, but it was more pronounced at week 4, at least what we saw in this particular study. And I think that's, again, you tend to see correlation, the timing at which, correlation with fluorescein angiography, the timing at which you see the effect is obviously, can impact this, and that's just the biology of the system. So again, we tend to see that both the angiography and these flat mounts do correlate with one another. We've also gone on to do some histology on these lesions. So what I'm showing you here is just a couple of representative images of these burns. So in BSS treated eyes, the energy was delivered here, as you can see, and we've tried to draw calipers of the choroidal layer, just kind of looking at the overall thickness of that layer, which is where the vessels reside, of course. And what we do find is that in BSS treated eyes, that area or region tends to be thicker than in ILEA treated eyes. We're currently actually doing some immunohistochemistry to try to characterize the cellularity of this, how much of this is macrophages versus choroidal vessels, new vessels. You can appreciate here there's some scarring going on in this particular animal. So we're doing a number of other things to help characterize this model and understand the biology, and hope to publish this within the next 2, 3, 4 months once we collect more data on this model. So in conclusion, what we found, the larger size of the swine eye is more relevant to the human eye, and I think that's a big advantage to this model. It really allows sponsors to formulate and put their compounds together in a way that they would do in the clinic, and that helps facilitate a lot of things, not only looking at a therapeutic effect, but how you dose, what kind of syringe do you want to use, how long do you hold the syringe in after you inject, and these sort of things. The model provides quantitative assessment by evaluating neovascularization by angiography and flat mount analysis. So these are 2 quantitative methods for evaluating new molecules. The model is very easy to reproduce and is economical. Because it tends to be a 2 to 4 week study, we don't have to house the animals for that long, fortunately, because pigs grow very fast. So it's something that we can offer that really is not going to break the bank. And characterization of this sensitive and translatable model allows effective evaluation and proof of concept of new treatments or delivery. So I'd like to thank those that contributed to all of this work. Dr. Brian Gilger, he's a professor of ophthalmology at North Carolina State University. He's our consulting veterinary ophthalmologist. He's board certified in toxicology and ophthalmology. See him every morning here, and he's just a great friend and collaborator. Christy Powell, she's our facility manager. Dr. Grazia Spiga, she's a scientist here at Powered Research with more than 10 years in experience in the eye. And Justin Prater, he's our senior project manager. He's got more than 15 years of experience in the CRO world.
For starters, Christine or David, I'll let you decide who starts, but can you just comment on how easy or difficult it is to use the Micron X system that you presented today?
Sure, yeah, I can comment on that since we have been using it quite a lot. I find the system to be very easy to use. Probably what I like most about Phoenix is equipment, besides the nice images that we're able to collect, is the user interface is very simple and easy to get through. I'm able to image a number of animals in a short amount of time, and obviously that's very important to me in getting high quality images. So I definitely am a fan of it.
And I agree. I've been using it on a number of different species, and once you figure out the positioning of the animal, it's quite easy to figure out how to image the eye. It's the same process. And as far as training people, since that's a big part of my job, the training of the Micron X has been incredibly easy. So it's about a 20-minute learning curve, and then it's just figuring out the light level and focus that you want for the animal from there. So it's quite easy to use.
Okay, perfect. David, a question for you. How many animals per group do you typically need to run to show efficacy?
Right, good question. So what we have found in our studies is that in a four or five animals is usually the best. Again, we went through IACUC approval to do these lesions in both eyes to help minimize and reduce the use of animals. We monitored the animals very closely for weight gain and just behavior, and they tolerated sort of bilateral lasering and treatment very well. And so with that, you get both eyes from one animal. So it's a lot of data, and we found that we can see significant results. Hopefully that's been demonstrated here in a very not very small, but a relatively small in these studies.
Okay, perfect. Here's a question that maybe both of you can answer, and actually these come in. So Michael has asked what type of export files are available for still and movies, and then Sabia, this is kind of related, has asked how you measure intensity of fluorescein. So do you use ImageJ or similar software? Christine, maybe I'll just start with you. Can you talk about the image formats that can be exported using the system?
Sure. So the movie, off the top of my head, I can't remember. I'm not sure if it's .avi, but the images are TIFF, which is readable. It's a raw image file, and it's readable by a bunch of different programs.
Okay.
Yeah, they are AVI files, and you can actually pull the TIFF files. Obviously, provide the highest resolution. They're bigger, but they're usually what I grab for the images. And I guess to follow up with the angiography, so yes, what most of the work for quantifying the calculated total lesion fluorescence, or excuse me, corrected total lesion fluorescence image, we use ImageJ, which is a freeware that probably most of you are aware of. You know, this is a method that we use that gives us a quantitative assessment. There are other methods of evaluating these lesions that have been reported in the literature in non-human primates where they do a qualitative assessment where they rank sort of the overall fluorescence, leakage, and just the appearance of the lesion and define certain criteria for that. And so that is another method of doing it. It's semi-quantitative, and you have masked people score them, and then you sort of compile that data. So those are two ways of assessing angiography in these animals.
Perfect. All right. Thanks for that response to both of you. David, how long do the lesions remain leaky? Would you have an answer to that?
Yeah, so we have looked in this particular model out to about a month. We've not looked longer than 4 weeks. In other CNV models that we have run here that sort of have a similar phenotype, what we have found is these lesions really persist for several months. And even what we have found in rabbits, that when we treat them with the Eylea drug, that they remain non-leaky for about 6 weeks, and then after that time point, 6 to 8 weeks, we find that these lesions then begin to leak again. So, you know, that's what's reported, similar to what's reported in the clinic, why patients have to go in every two months to get another shot or another treatment. I would suspect that it would be similar in this species, but those are studies that we have planned in the future.
Okay, perfect. And how many animals can you run at one time during a study?
Right. So as I mentioned before, we usually run about 4 per arm, and you know, really as a function of space in the facility and sort of throughput. Our largest study here at our facility has had 30 pigs in it, so we had 7 arms, and what we did essentially was broke the procedures up into 2 days. Simply from a you know, a feasibility standpoint, recovering 30 pigs at once is not an easy task without an army. So we you know, again, we try to just do about, you know, 12 to 14 animals a day, and that works really well. And again, that allows for, you know, 3 arms for a dose response. You have your controls, and maybe you have you know, a synergistic group where you're putting EYLEA in combination with your test article. So those are the types of studies that we tend to see and run here.
Okay, great. Dr. Zhao has asked how one goes about injecting the fluorescence, I guess, for the fluorescein angiography. Christine, do you want to maybe start by answering that question? He specifically asked if it's done through IV injection.
Sure. Yes, for the non-human primates, it was IV injection, so similar to a human. It's just right in the crook of your arm, so it's an arm injection, so you have to have the IV all prepared. But the nice thing about this is that you can push in the fluorescein and start the timer exactly when it's been injected into the animal, and you can see it come into the animal, whereas with the mouse doing the IP injection, it can be a little dicey repositioning them.
Okay.
Right, and I can follow up in the pig. What we do is, as I mentioned, we will place a catheter as well, similar to what I'm sure they do it in the non-human primates. But we do that in the ear, actually. So the ear vein is, or ear veins, much like in rabbits, are very easy to find and hit in the pigs. So we'll place a catheter to be able to administer the fluorescein, and it is intravenous, and it's, again, you can start the clock as soon as you inject, which is what's very nice about this. Your times can be very tight, which is important in angiography.
Okay, great. We've had a few questions come in. Specifically, Rajendra asked about the power of the laser that was used or is generally used for mouse research and also large animals. So can you comment, Christine, maybe I'll start with you about that. And in general, is there a standard for optimizing the system when it comes to, you know, the different parameters that are used for laser, duration, power, etc.?
Sure. So we make a laser attachment to the Micron 4, and that's for the mice and rats. So I can't speak for the large animal lasering, but I think David can. But as far as the smaller animals go, we have a standard recommendation for our specific laser and our specific setup, and then from that, you really have to calibrate the system specifically for what you want to do. So some people prefer slightly stronger lesions, and others would like to do a lower level. And so it depends really on what laser you're using with the large animals and how your specific system, the optics, are set up. So it's really important to calibrate it. So you can ask for the general recommendations, and then from there ask for more specific ones. And with the Micron 4 laser, you can contact us at any time and we can help you out with that.
Okay. David, how about you? What's your experience with
So,
setting up all these parameters?
Right. So what we use, as I mentioned in my talk, we use an 810 nanometer laser, and that is fired through an indirect ophthalmoscope. So it's a little bit different than what you might use in a rodent model. The Phoenix system, again, has a very nice sort of laser-guided setup that really allows you to sort of dial in, you know, the spot that you want to target in the rodent, and you really can modify spot size and, again, duration of the burn and the intensity at which you do burn. So even within each type of laser, so there's a 532 laser, which is a green light laser, and then the 810 that I'm using is in the infrared spectrum. And so what you want to do essentially, as Christine highlighted, is really optimize it. So in our case with the pigs, we will fire through an indirect, and our power range is anywhere from, say, 200 milliwatts up to 500 milliwatts. And again, these are experiments that we did very early on where we sort of, ramped up the intensity and the duration of the burns. They can be anywhere from half a second up to, a second and a half. So within that range, you've essentially just got to experiment with that. You've got to look at them. You've got to see what gives you the phenotype, and more importantly, you've got to see whether you have a therapeutic effect. And that's what's beautiful about this model, is that we've got a great drug, EYLEA, to be able to evaluate, okay, is the model working? It's a great positive control because we do see inhibition, especially with the parameters we've set up. But everyone's laser is going to be a little bit different. Everyone's indirect ophthalmoscope is going to be a little bit different, and probably everyone's Micron is going to be slightly different. So it's really an optimization step that you've got to take when you want to do these experiments.
A question for you, David, I guess to start with is, do you think that the mini pig eye and the swine eye are different? And I don't really know in what context this question is being asked, but
right.
Perhaps you can just comment on that in general.
I think they're probably very similar in size. So the mini pigs are, obviously strains like the Göttingen and some of the other ones. They're not coming to my mind right now, but they're obviously bred, to sort of remain small so that you can do sort of longer duration experiments on them. The eyes are very similar in size and in their physiology. So I think that you would see very similar results in our hands, in mini pig eyes versus, weanling pigs from just standard farm pigs that are young animals.
Okay. Christine, do you have anything to add to that based on your experience?
I have not imaged mini pig eyes, so I do not have anything to add. Thank you, though.
Perfect. All right, one last question for you, David, that's specific to your talk, and Sabia would like to know what's the concentration of EYLEA that you inject into your animals?
Gotcha. So in this case, we did give the same therapeutic dose that's been given to patients. So it's the 2 milligram dose.